This is my first, this is the only video on heme metabolism. I hope to cover it all in one video. The learning objectives are to describe the steps in heme synthesis and its regulation, list the two steps in heme synthesis inhibited by lead, tell how acute intermittent porphyria differs from Porphyria, Cutanea, Tarda. Here are blisters from Cutanea, Tarda, and here are the UV light from Cutanea, Tarda. The urine samples, here's normal and here's red. It glows red under UV light, and these people have a lot of spots on their skin where the intermittent does not have that problem. Describe how heme is degraded. Critical products and intermediates. Described how bilirubin is transported in the blood and the difference between conjugated and direct reacting bilirubin. Tell the causes of jaundice in adults and newborns and its basic treatment. Heme is essential. We need heme for hemoglobin. We need a little in the muscle for myoglobin. We need it especially for cytochromes, for oxidative phosphorylation to synthesize ATP. We also need it to metabolize drugs, P450. We also need and it's in catalase. There's heme's in that as well. So heme is a really important molecule in our body. And we, for red blood cells, for example, we have 25 trillion red blood cells. They live for 120 days, so 200 billion are dying and being remade every day. 200 billion. That's 6 grams of hemoglobin a day. Haptoglobin binds hemoglobin when the red blood cells die, so the hemoglobin doesn't get lost in the urine. Then hemopexin binds heme, so the heme doesn't get lost and doesn't get out in the urine to save the heme and keep the iron because we want to conserve iron. Transferrin binds Fe+3, and then ferritin stores Fe+3 inside cells. So we have a storage form. So we have 4 proteins that are involved in making sure we don't lose iron, because generally iron's hard to get. Now, Protoporphyrin 9 synthesis. So it starts out in the mitochondria with glycine, which is a non-essential amino acid. Since something like heme is so important, it's critical that it use non-essential amino acids rather than essential amino acids. It also uses Succinyl-CoA, which is a TCA cycle intermediate. There are 2 enzymes that catalyze the very first step to produce delta aminolevulinic acid, or ALA. The first enzyme, ALAS1, is found in both the liver and the bone. The second enzyme, ALAS2, is found only in the bone, and it is primarily responsible for making heme for red blood cells, for hemoglobin. Both enzymes use pyridoxal phosphate or vitamin B6 as cofactors. Then, so we make delta aminolevulinic acid. Then that's acted on by ALA dehydratase in the cytosol. So it starts out in the mitochondria, then moves into the cytosol. In the cytosol, that goes to porphobilinogen. So porphobilinogen is this product here, and if you have lead poisoning, and some children live in houses where there's old lead paint is one of the factors. We used to have lead in our gasoline. That's been taken out. But if they live in a house with old lead paint, or they live in Flint, Michigan, they might very well have too high lead levels, and they can't make this compound, so they are anemic. Then the next step is the porphobilinogen is converted into uroporphyrinogen 3. Uroporphyrinogen 3 in the cytosol, and this, if this cannot happen, we have a condition called acute intermittent porphyria, and we'll talk more about that later. If the next step, this compound, the uroporphyrinogen 3, can't be metabolized on down to protoporphyrin 9, then you have a disease called Porphyria cutanea tarda. This is the most common porphyria that there is. This one's right behind it in terms of so ALA1, liver and tissues, heme synthesis and gene transcription. ALL2, so heme feeds back to slow the gene transcription of ALS1. ALS2 is bone, transcription is upregulated by erythropoietin, and translation is upregulated by iron. And the cytosol, the second step, is a non-competitive inhibitor by lead. Basics. This is succinate and glycine, so this is the structure of ALA. Do you need to recognize that? No, you don't need to remember that. I just thought it'd be interesting if you saw it. Sideroblastic anemia is a defective ALAS2, an immature nucleated red blood cell with ferritin iron granules that accumulate within the mitochondria. The granules often form a ring around the cell, sort of like this diagram here. Picture is not a true cell. Here's a real cell here, and can be seen in the erythrocytes as well. They are seen in peripheral blood smears. Left shifts suggest anemia. So when you see those, Dr. Paul Monaco provided me with this information. You'll see more about this when you get to cell and tissue, but this is sort of an introduction of it. The final step is adding the iron into the protoporphyrin 9 ring. And this one occurs back in the mitochondria, and it requires the enzyme ferroketolase, and it's also insensitive to non-competitive inhibition by lead. So lead binds to a site other than the active site to inhibit this one as well. So lead's a real bad actor in heme synthesis. And lead poisoning in heme synthesis is, I said, non-competitive. It's the second step and the last step. It results in a microcytic anemia. Micro, the red blood cells are smaller than normal. They're also hyperchromic. They're paler than normal. You can look right through them. Here's a lymphocyted comparison. So the step one question you quite often hear about is a child living in a home with lead paint, or in Flint, Michigan now where they had lead in the water. So here is, again, remember about non-competitive inhibitors. So here's the line the Michaelis-Menten plot for non-competitive inhibitor. It changes Vmax but not Km. Here's the Lineweaver-Burke plot. Km does not change, Vmax does. Acute intermittent porphyria. Acute intermittent porphyria is mainly a liver disease, and ALA and porphobilinogen in the urine. So you see the initial product and the porphobilinogen in the urine. It's not photosensitive. This compound is just a single ring, so it doesn't absorb UV light. It's not conjugated like this one is. This one absorbs light. So these people don't mind being out in the sun, so they can work outside. However, their condition is exacerbated by drugs and alcohol, because this is made in the liver where P450s are made. So if they can't make enough P450, their P450 is not functional, then they have a lot of trouble metabolizing drugs and alcohol. They also have neurologic or psychiatric issues, usually seizures and pain. It is thought that Mad King George III, who was the King of England during the American Revolution, and Vincent van Gogh may have suffered from acute intermittent porphyria. The other one is Porphyria cutanea tarda. It's a chronic and most common form, and these people, it affects both the liver and the bone. They wind up with this product uroporphyrinogen 3, which is right here, uroporphyrinogen 3 in their urine, and they are photosensitive. They don't like being out in the sun, so they tend to go out at night, and they because they get spots on their skin, and they'll also have their teeth will almost glow in the UV light, and so they were referred to quite often as vampires or werewolves. Very not a pleasant disease, and they should never be made fun of. Urine has this coral red color under UV light. You can see down here in the Cutanea tarda. The acute intermittent also has their urine will also turn a dark purple on standing. So note these differences in these two. This one will eventually polarize to a dark purple product. Here's an overview of the porphyrias. Acute intermittence, the first one in the pathway. Remember, Succinyl-CoA and Glycine goes to porphobilinogen, and this is the one that you get in acute intermittent. Then Cutanea tarda is winds up having an excess of uroporphyrinogen 3. You can't metabolize this any farther, so this builds up in the urine. So you have this one in the urine with acute, and this one in the urine with the Cutanea tarda, and so you can't make enough heme. So they're both of them are nasty diseases. And again, this tells you that iron stops the last step and the second step. Heme degradation and excretion products. Remember, we're making 6 grams of hemoglobin a day and we're turning it over, so they're being turned over rather rapidly. Liver and macrophage are mainly responsible for this, so when the red blood cells die, the macrophage phagocytize them, take them in, digest them away, and we make biliverdin first, that's green, and we make carbon monoxide. It's very little carbon. This is the only step in the body where we make carbon monoxide. A little of that carbon monoxide damages our hemoglobin, but not much. Bilirubin then is the next one, and it is red-orange. We make 300 milligrams of bilirubin a day. And bilirubin is not soluble, so its only way it can be transported in the blood is bound to albumin. It gets transported by albumin. That's one of the reasons we have so much albumin. And goes to the liver, where it's conjugated with diglucuronide. So we put sugars on it, so we make it soluble. So here, the bilirubin diglucuronide structure, you can see the sugars. This is very hydrophilic. This is very hydrophobic. This makes this water soluble. And then this can be secreted in the bilirubin diglucuronide goes into the bile out of the liver and goes into the intestines and can be metabolized. So we have biliverdin, bilirubin, albumin, bilirubin goes to the liver where it gets conjugated and then goes out. Jaundice refers to yellow, also called icterus, and hemolytic jaundice, red blood cell hemolysis. If for some strange reason you're lysing a lot of red blood cells and they're dying so fast that the heme cannot be degraded, then you'll wind up with jaundice from that. And you'll have normal urine and normal brown stools in that condition, but you'll still have jaundice, which is not a good thing, and so you're having to make a lot of new red blood cells as well. Hepatocellular jaundice, that's where you have liver damage. So the liver can't conjugate bilirubin fast enough, so the liver's lost its function. Liver cirrhosis from alcoholism, various other drug problems can cause this, or even liver cancer. Dark urine and normal to pale stools. So they can still, the urine has can secrete the urobilin, so you wind up with dark urine, but you can't you don't get it into the bile, so you wind up with very pale stools. So, and the doctors use that to see what's wrong with you. Obstructive jaundice, bile duct obstruction, gallstones, and pancreatic cancer. Again, you get dark urine and you get pale stools because if you have gallstones blocking your gallbladder, it doesn't let the bile flow into the intestine, so you don't get your bilirubin into the intestine for metabolizing it. Pancreatic cancer can also close down the bile duct, and so it doesn't come through. Jaundice in the newborns. Immature livers can't conjugate bilirubin, and so they wind up with immature having less of the enzyme glucuronidase transferase, because the liver, that's where it's made, that transfers this. And so these children have higher levels of bilirubin. They put them under UV light, and the UV light oxidizes the bilirubin to water-soluble products, and they cover their eyes to make sure you don't damage their eyes by the UV light. Bilirubin measurements. Conjugated or direct reacting bilirubin is the water-soluble fraction. That's the conjugated form that's in the blood. Total equals conjugated plus unconjugated. Usually that's done by a methanol extraction of the blood plasma. So to get the unconjugated, the form that causes the deposits in the skin and eyes and under the tongue, you take the total minus the conjugated. Those are the two things we can measure, and you get the one we can't measure very well because it's insoluble, and that's the unconjugated. So the unconjugated is the one that causes jaundice. Here's an overview of this whole process. The red blood cells die, macrophages take them up, and those macrophages are usually in the spleen and liver. Then the bilirubin is bound to albumin, taken to the liver where it's conjugated. The conjugated bilirubin is secreted in the bile duct where it goes into the intestine, and there it is converted to urobilinogen, and some of the urobilinogen gets retaken into the portal vein, goes into the blood, and the kidneys filter this out and convert it into urobilin that's seen in the urine. The rest of it goes to eventually bacteria metabolize it to stercobilin, and that winds up in the feces. So now you know why feces are brown and urine is yellow, because this is what happens when you metabolize bilirubin. And this is a picture of me with Ivan the Terrible in London a few years ago at a night at the Museum 2 premiere street party. It is better to travel and hope than to arrive. Thank you. Bye-bye.